Okay, this is the Professor, CBMD PhD. It's today Thursday, September 14th, 2023. It is 11:16 and 13 seconds, a.m. Eastern Standard Time. And today's research, myeloma. So let's hop to it here. Mechanisms by which multiple myeloma escapes targeted, immunotherapy identified. International research team says findings may help screen for vulnerable patients and lead to new approaches for future therapies. Date August 31st, 2023. Source: University of Miami. The targeted T cells can be rendered useless if the antigen they are tracking mutates, essentially disappearing from the radar screen. Here the researchers report mutations that thwart immunotherapies engineered to seek out two targets in multiple myeloma, allowing previously treated cancers to adapt, escape treatment, and relapse. The scientific investigators and microbiologists say that recognizing these mutations and gaining a clear understanding of the resistance mechanisms to these potent immunotherapies is pivotal. This knowledge plays a crucial role in devising tailored strategies and making informed choices regarding the selection of products and targets for individual patients. Antigenic drift is well established as a mechanism that tumors employ to develop resistance to immunotherapy. In this study involving 30 patients, the researchers focused on changes occurring in two potential targets on plasma cells. The B-cell maturation antigen, BCMA, a G-protein coupled receptor family C, group 5-member D, GPRC, 5D, chimeric antigen receptor, T cells, or CAR T cells, and bispecific T cell engagers. It is called TCE or immunotherapies that can be engineered to target these antigens that have shown promise in treating relapsed or treatment-resistant multiple myeloma. Over time, however, and as the cancer cells change, effectiveness often wanes. Few studies have been done to identify the reasons for this diminishing clinical response, and this is believed to be the first genomic study of intrinsic mechanisms of cancer cells that permit antigen escape in patients who relapse after undergoing these immunotherapeutic approaches. One important aspect of the findings is that mutations in the extracellular domain of BCMA have a distinct impact. They impede the binding of the therapeutic T cells without exerting any influence on the protein expression or downstream signaling pathways. This finding revealed that the proportion of patients with myeloma who experience relapse due to antigen escape is considerably more extensive than initially anticipated. The discovery of these mutational events that participate in multiple myeloma relapse underscores the critical need to screen patients for these variants, according to the article. Additionally, detailed characterization of these binding interactions will permit the rational design of next-generation T-cell redirecting agents and inform the optimal sequencing and combination of these immunotherapeutic approaches. And this is a Professor of Microbiology, Bioinformatics, Pathophysiology, and Nature of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.